And good morning, everyone. It is Thursday, molecular genetics time. So we were going through some old-fashioned genetics and talking a little bit about mutagenesis and the idea that genes are associated on chromosomes and they can be linked, tightly linked, or they can be on different chromosomes, in which case they are not going to exhibit an association, and that's really what independent assortment is about. And I thought what we'd do is carry on a little bit more in our discussion of mutagenesis and get to where we can think about what kinds of mutations there are, what makes a mutation, what is a mutant. And I thought what we'd do was we went through the idea of the complementation group, which is a subtle but really important point about thinking about genes and carrying out mutagenesis screens. So I thought what we'd do is go through a little bit of a classic Drosophila fruit fly mutagenesis screen. I'm going to put it in my pocket here, pardon me. And think about how we can go and look for specific mutations and actually then begin using mutations to analyze the gene and cloning, things like this. And also exhibits so we can determine a little bit more about a complementation group. So flies are a really good model system for doing mutagenesis because they only have 3 chromosomes. We've got a lot more chromosomes. We have 23 chromosomes, and the fly has got 3. There's a little chromosome 4 that doesn't code for anything. And because of the polytene chromosomes, you can actually visualize them, but you can also begin to carry out mutagenesis screens using a variety of different techniques. One way to carry out a mutagenesis screen is just to mutagenize the flies and then begin carrying out back crosses where you are making the resulting progeny homozygous for a mutated chromosome. And this is what you would do if you're doing just a genome-wide screen. And that's not what we want to talk about. We want to talk about a directed mutagenesis screen. And it's something that you can do particularly well with flies. Turns out you can actually kind of do these things with humans, but not in a direct way. This is using something called a deletion chromosome. So by using X-ray mutagenesis, you can generate chromosomes that are quite abnormal, that have lost a chunk of DNA. They actually have undergone a deletion. And you can visualize this actually in a polytene spread. You can begin to identify it by a variety of different ways. But we can, in the Drosophila field, we have strains that are carrying deletions up and down every chromosome. Now, pretty much the way we think about a deletion, we're gonna just for a second look at chromosome 3 for no other reason than it's a chromosome that I did a lot of work on. And here is the diagram of it. All right, so chromosome 3 is broken up into 2 segments, 3L and 3R, left and right, and you typically would work on just one side. But the chromosome is going to look like this. Now, you can identify and find deletion mutations, and these are going to be deletions of several million bases. And, in the parlance of the polytene chromosome, they're going to be missing specific bands. So you might find and identify a gene in a specific area of the genome, and this can be done by a lot of different ways, but let's just say we're going to mutagenize a specific spot on the chromosome. You identify a deletion chromosome, and so in the heterozygous state, or I guess in the diploid state, it's going to look in a graphic like this, where we've got the deletion chromosome is missing a piece. Now, the chromosome itself is not in 2 separate pieces. It is joined, but it is physically missing this piece. It's a shorter chromosome. And if you've got genes in this deletion region, what you've created is a state that is known in the biz as being hemizygous, in that they're not mutant or missing all of the chromosome, just a portion of it. And another sense of doing this is we've uncovered this region. We have rendered this specific region of the chromosome single copy. And so now we can pick up recessive mutations by screening for mutations using the deletion or the deficiency chromosome. So what we're going to do is take a chromosome that's got a couple of visible markers on it so we can actually identify it. Typically these would be dominant markers that will not be lethal but will produce a thickened bristle or a change in a patch of bristles or a wing shape. So you take males, mainly we do this with males because we can generate a lot more mutant sperm than you can generate mutant embryos or mutant eggs. And you're going to mutagenize them. A standard mutagenizing agent is ethyl methane sulfonate, abbreviated as, of course, EMS. And ethyl methane sulfonate causes point mutations particularly. It will cause individual nucleotide changes. And it generally doesn't result in deletions, in single nucleotide deletions, which would generate a frame shift or some kind of thing. It is going to be a nucleotide change. So you can go from an A to a T or a G to a C, things like that. All right? So actually, it's not all that critical that we know. You go from you do a purine to purine shift or a pyrimidine to pyrimidine shift. All right. So you're going to change the individual nucleotide. These are going to be fairly subtle and delicate mutations, and you would typically take 50 or so males, put them in a bottle with no food or water for a couple of hours, kind of get them thirsty, and then dump them onto a paper towel soaked with ethyl methane sulfonate and a sugar solution, and allow them to sit there and feed on this overnight. Now, the next day you come in, you take those males, and some of them probably croak, but most of them are still okay, and then you mate them with a big harem of females. Now, the females also have specific markers on them, and actually the way it works is fairly convenient for the females to have a number of different kind of marker chromosomes. And from here, the females probably are also carrying the deficiency, the deletion, and you begin looking for the flies, the progeny, that are carrying the mutagenized chromosome, and you know that by its marker, and the deficiency chromosome. So you've done, with the EMS, you've done a whole genome mutagenesis. So you've mutated all kinds of stuff, but you're only looking at this very specific spot. So let's just imagine this. We have mutagenized all up and down. We've made hits all up and down this chromosome because we really EMS'd them pretty hard. Can you see my little red marks there? Fine. Now, if we were to take this chromosome and have it in the homozygous state, you would have lethals and you wouldn't be able to tell what's going on at all. They would just be dead. But by placing them over a deficiency chromosome, this little deletion, mutations that are all here and mutations that are all here are not going to be seen because really most mutations end up being recessive mutations. Dominant mutations are phenomenally rare, and there are a lot of genes that can't even generate a dominant mutation. That's just not how they work. But recessive mutations, that can happen pretty easily. So what you're doing is screening for mutations in this region. Now, depending on the organism that you're working on, you can calculate the more or less average amount of space per gene. It turns out in a fly, there is approximately one gene per 10,000 bases. For humans, it's a little bit bigger. It's probably one gene for 50 to 100,000 nucleotides. So we got a lot of space there. So one gene in 10 kb. So if you've got a 300 kb deletion, you might expect that there's going to be 30 or so genes in there. And what you're going to do now is start carrying out pairwise mutagenesis analysis, where you're actually taking individual chromosomes, mating them with a deficiency, and you're looking to see, is there some kind of phenotype? And very often you're looking for lethality. Or if you've got some kind of very specific thing, I actually did a mutagenesis screen where we were looking for a crooked leg, and so we were screening everybody. And what you end up with is something like this. So you've got a variety of different marked chromosomes. We're going to just go ahead and call this chromosome 3 with the asterisk, meaning that's our mutagenized chromosome. We're going to put this, make this, this is going to be over, or the other chromosome pair is going to be a wild type chromosome 3, but with some kind of dominant marker that we can visualize, and it's also going to have a recessive lethal. So let's just call this TM6B, which is third multiple 6B. It's a standard chromosome 3 mutagenized chromosome. It's carrying a recessive lethal mutation. It also has some visible markers. All right. Now, we're going to cross this with our deletion chromosome. And this is also over TM6B. Now let's look at the resulting progeny from this. We're going to have, and we're going to just go through and do a pairwise pairing up of the individual chromosomes. So you got the different gametes that are possible. And it ends up looking like this. We're going to have mutagenized 3 over the deletion. We're going to have mutagenized 3 over TM6B, our marked chromosome with the recessive mutation. Now we're going to have TM6B over the deletion. And we're going to have TM6B over TM6B. So this is a standard cross now. And let's look at what these things are going to be showing us. If we have generated a lethal mutation that is uncovered by the deletion, this guy is going to be lethal. This guy is going to actually be our recovered stock. So if we do have a lethal mutation, we can actually grow it up from these guys. This guy is going to have a set of markers that are associated with the deletion and TM6B, so we can identify them. We have to pull them out, but we can identify them. And then lastly, we will have that. And because these two chromosomes are carrying recessive mutations, as a recessive, it's going to be now lethal. All right, so when we look at this, there are a couple of tools we have for very rapid screening of thousands of individual crosses. Mainly, with TM6B, there's a visible marker. The way the larvae are shaped, they actually are, rather than being sort of long, pencil-shaped larvae, they're almost football-shaped. They're like squished down. It's a mutation called tubby, and it's a dominant mutation that is really visible. And so when you go through this, if you have a lethal that you've uncovered here, this one won't be carrying TM6B, it won't be squished down. This one is going to be tubby, this one's tubby, and this one's lethal. And so you just take your vial of flies, hold up the light, and you look to see, do you see any long, normal-shaped larvae? If you do, you didn't have a mutation there. And it's not lethal. If you have no normal-shaped larvae, then that means that this guy is dead, and you want to pull this one out and start growing it up. And so you would carry out a mutagenesis screen, several thousand individual chromosomes, individual crosses like this, and then you begin doing complementation analysis. So let's say with a mutagenesis screen of using a deletion that is 300,000 nucleotides, 300 kb. You might expect to have 30 or so genes in there. You're going to start picking up mutants. You don't know what genes they're associated with. Once you have a series of lethal mutations, you then begin pairing them up to see if they complement or fail to complement. And now remember, the idea is with complementation, if they're in two separate genes, we're going to have complementation. If they are in the same gene but two separate independent mutations, they will fail to complement. So once you've established your complementation groups, that's how you begin to start organizing things. And a statistical sort of rule of thumb is you can say that you have mutagenized pretty much everything that there is to be mutagenized if you can demonstrate that every complementation group that you've generated has more than one member, because that's one of the things that can happen as you're carrying this thing out. You'll have a complementation group that is a mutation that is a single fly strain. You only got it once. And that means that you probably statistically haven't saturated that area as well as you could. So you keep on mutagenizing, and then after a while your advisor says, okay, you can stop after a few years of doing this. So this is a standard way to carry out mutagenesis. Now, what you're doing is creating recessive mutants. And let's look to see what kind of things we can generate like this. So, in terms of mutation, these are the things that you might do to the DNA to generate a mutation. You can have a deletion in which you actually have lost multiple nucleotides. You can have a single nucleotide deletion. So we're going to say this is more than one nucleotide, and this is a single. Then we can have a nucleotide change. So now within this kind of mutation type, we have a couple of things that can also be happening. We can have a mutation in the promoter region. We could have a mutation that results in a frame shift. We could have a mutation that occurs in an intron-exon splice junction. Now, with a promoter enhancer type mutation, this is going to affect the amount of gene product being generated. And it could potentially affect where the gene is actually being expressed. That's probably fairly rare, but it can certainly happen. You can mess the promoter up such that it doesn't work anymore. Now, much more common is the kinds of mutations in which we have single nucleotide deletion, in which we can generate a frame shift. So a single nucleotide mutation within the open reading frame, that is, within the portion of the RNA that is actually being translated, is always going to be a bad deal, because when you frame shift, the amino acids after that frame shift are going to be completely different than what they should be. And very often, when you're in the wrong reading frame, we'll be looking at some translations here probably next week with a little assignment. When you're in the wrong reading frame, it's going to be stop codon after stop codon after stop codon. So if you're in the wrong reading frame, it's garbage. Now, a nucleotide change is going to be a bit more subtle, in that a nucleotide change could generate an amino acid change. Now, when you look at the codon charts, the codon chart, as we start to look at it evolutionarily, is designed to actually kind of handle this. That there are a lot of codons that you can change one or two nucleotides and not change the amino acid identity. There are some that if you change a nucleotide, you change the amino acid that is being called in. Often the amino acid is going to be a similar biochemical amino acid. It'll be a neutral substitution. But there can be mutations that occur that will generate a different amino acid that results in a different kind of protein shape. By and large, when you're looking at lethal mutations, you're probably looking at some kind of either a frame shift mutation or a splice junction mutation. Splice junction mutations, you can have, when you have the exon intron splicing, there are invariant nucleotides on either side of the splice. You mutate one or both of them, one of them, and you don't get the splice. And now you've got the open reading frame. You should have the intron spliced out. It's not, and the introns are going to be filled with stop codons. And so it effectively is a frame shift type mutation, or an exon intron splicing mutation is going to result in a stop. So that's generally the kinds of mutations you get, is something that's going to prevent the protein from being able to actually be translated. So these two kinds of mutations are typically known as point mutations. Now, a point mutation is going to be a single nucleotide, and as we said, if it puts in a stop codon, changes the nucleotide to make it a stop codon, that's going to be deleterious. If it is going to be mucking about with a splice junction, that's going to be deleterious. Changing an amino acid might not do anything at all, but probably not going to be as common. But you can hassle with that. So what we end up with then is mutations that fall into a pretty defined set of categories. And we can really begin to learn a lot about the way a protein is functioning by doing mutagenesis analysis. So if we kind of just sort of list the sorts of things that can happen to a gene with mutation, we end up with this. We can have a frame shift. We can have an exon intron mis-splice. We can introduce a premature stop codon, or we might have some kind of effect gene expression levels. By far, the most common things are going to be exon intron, missplice, stop codon. Depending on the mutagenesis that you're using, you can have a deletion that will result in a frame shift. But for the most part, we're going to end up with single nucleotide changes. Now, a term that is used in the mutagenesis biz is this. It's known as a single nucleotide polymorphism, more colloquially known as a SNP, an SNP. And you see that? My marker's starting to go on me. An SNP. Now, a single nucleotide polymorphism better. A SNP, this is a wonderful tool for thinking about mutagenesis, single nucleotide polymorphisms. Now, one thing I will say is that of the kinds of mutations that you could generate, single nucleotide polymorphisms by far are the most difficult to actually detect at the molecular level. Deletions are pretty easy because you're actually going to have changes in size of the DNA that you're working with. Single nucleotide polymorphism is just a single nucleotide change. And this gets into an interesting philosophical question, and that if we were to sequence our genomes, we would find that we are 99.99% identical, but we are going to have arrayed throughout the genome single nucleotide differences, these SNPs. Now, it turns out that when we start thinking about the population and the differences within the population, these differences all come down to single nucleotide polymorphisms. This is what companies like 23andMe are doing, is they are identifying single nucleotide polymorphisms within a genome, and they have a database that they're constantly building that is things that are associated with this single nucleotide polymorphism. And so it brings up the point about what is mutant and what is wild type. So I would argue that if you're, 18, 20, whatever your age is, and you're sitting around, you're wild type. You made it. Congratulations. I guess if you have children, you gotta have children regardless. We can have a huge range of nucleotide differences and still be considered, quote, wild type. And it's what's really interesting to think about in terms of evolutionary genetics, population genetics. What is the standard gene set? The standard gene set can be actually a range of things. As long as it works well enough, you're good. Because single nucleotide polymorphisms are what is driving evolution. But at the same time, we have within the population a whole bunch of single nucleotide polymorphisms that are completely functional. So I often think about, sort of the role of mutagenesis in thinking about populations and evolutionary changes that are occurring through single nucleotide polymorphisms. Now, you start to have chromosome rearrangements and things, and when you begin looking at a mouse genome versus a human genome, we can actually have correspondence between the different chromosomes, and they've actually undergone chromosome rearrangements, but the genes are all there. And yet, there are still single nucleotide polymorphisms that have resulted in the difference between a mouse, squeak, squeak, and myself. Hi. So, single nucleotide polymorphisms are a really important tool for thinking about mutations. Now, prior to the ability to sequence genomes routinely, there are a number of ways that mutations were identified. So now let's think about this. We've got, and I'm just looking to make sure I'm keeping up more or less with my chapter, blah, 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 blah. All right. So we have identified a gene of interest, and there can be a number of ways you can actually clone the gene of interest. Sometimes you have sort of in flies, you can work it from a number of different directions. You might have a gene that you've identified, and then by doing what's known as in situ hybridization, where you take, say, your gene of interest, like a cDNA that you identified, and you label it up. And back in the original days, we used to label it up with tritium, and you take a chromosome squash of the polytene chromosomes, so you got X, 2L, 2R, 3L, 3R. And you take your DNA, you make a probe, not with P32, because the particle is too energetic, but with tritium. And you take a chromosome squash, you dunk a little bit of sodium hydroxide once you've squashed it, and then put it into a salt buffer. The sodium hydroxide, of course, is going to break apart the hydrogen bonds, denature the DNA. You take the tritium-labeled probe, boil that up, and then you actually physically place the probe onto the microscope slide, cover it with a little bit of parafilm or some way seal it up, and let it sit at 65 degrees or so overnight and come back and wash it. And with tritium, you then take this microscope slide and dunk it into photographic emulsion. These are things that people don't even know about anymore, but we used to take pictures using a silver grain based material on a negative, and the silver grains were actually suspended in gelatin. And so you'd warm the gelatin up in the dark room, you go in, dunk your slide in there, pull it out, let it cool and dry, put it into a little box in the refrigerator, and let it sit and expose for a couple of days, and then you develop it. And what you would then see is a spot that has a series of silver grains on it. And so when you magnify this spot up, you have the bands and interbands. And then you have a spot that is showing an array of silver grains exposed, and that is the spot where your probe is hybridized. And you then take out your CRC book of the maps of Drosophila and start trying to figure out, all right, what chromosome is this on? Typically what I would do is I'd go across the street to Baylor College of Medicine, where my friend Juan Botas could sit and read my chromosome in 15 minutes and go, oh, yep, that's probably 3L band 71C. I was like, okay. So if you stare at chromosomes long enough, you actually can be able to read them. And what you have now done is identified the specific location. So you go back to the lab and you look around at some of the books and see, are there any mutations that have been mapped to that region? And if there are, you call them up and say, hey, I'd like to see if my gene might be some of these mutations you have. So that's one of the ways you can begin doing this, from the gene to the mutation. Now, if nobody has any mutations there, you identify a deficiency chromosome around this spot, and you begin carrying out mutagenesis screens, and you start looking for complementation groups. So once you've got a set of mutations and you've got a gene, then what you're going to start doing is cloning the gene from those different mutations, those different mutants, and sequencing them and looking to see what kind of single nucleotide polymorphism do you see, and is it something that's going to actually affect the gene product? Is it going to affect the splice junctions? Is it going to be a stop codon? If it's going to be a nucleotide change that results in an amino acid change, you have to ask, is that amino acid change significant? Is it a dramatic amino acid change, or is it going to be a subtle shift? And so you have to do a little bit more analysis of that. Hopefully, you're going to generate a stop codon or a splice junction change that's going to result in a stop codon, and then you know, okay, that's where the mutation is. Now, the other side of this can be, that's if you've got the gene first, and now you're going for the mutation. But, of course, there are a bunch of mutations that were generated prior to the age of molecular genetics, and so you have a mutation, but no gene. That becomes a bit more of an interesting problem, because you've got a mutation, and it can be a really cool mutation. I really want to know what it is that is changing a shift in wing identity, or in segment identity, or a mutation that is affecting the learning and memory of flies. That becomes a bit more difficult. The thing you would do is, through a series of crosses, you can very quickly identify what chromosome it's on. And then by doing recombination analyses, using the recombination data that you can generate to make a recombination map, and we talked about that last time, is that you can map the chromosome based on recombination events, and you can actually map genes based on their recombination frequency. And it's not a particularly accurate way of doing it, but at least it gives you an idea. You can identify the region where your gene is. And by very careful recombination mapping, you can get it down to a fairly narrow region. Hopefully, someone's done some cloning in that area, and you can get a little piece of DNA in that area. That's probably the best way. There was almost a mythic way of taking polygen chromosomes and taking a teeny little glass scraper and scraping away a little piece of DNA and using that to start and build a little library of that region of genomic DNA. I guess for this, we'd like that big. And you make a little piece of it. I'm not sure how common that really was ever done. It was written up in book chapters, but it was really very tedious to do. More likely, you're going to have some piece of genomic DNA that you can identify very near your area. And you can begin something known as a chromosome walk. Now, first off, in the age of genome sequencing, the fly genome has been sequenced a lot. It was the first complex eukaryotic genome to be sequenced. And you can now go to the web and identify the genes, identify sequences, and you can basically do my entire PhD with a few clicks on your web browser. But the way you would actually go out and begin to identify these regions in the beginning was like this: carrying out something called a chromosome walk. So we've got a region of the genome that we're interested in studying. And who knows where your gene is? We're going to have to figure that out in a moment. What you can hopefully do is get a little piece of DNA from this chromosomal region. And so we got a piece of DNA right here. All right. Now you begin screening genomic libraries and identifying genomic fragments. And what you're going to try and do is find a piece of genomic DNA that is going to be like this. And you then make a probe from this end and try to extend your chromosome walk. Then you get a little piece of DNA from this end and extend the walk a little bit further. And each one of these clones is probably about 50,000 nucleotides, and you just keep on going. It is a very tedious process, and what I'm showing is an ordered set of clones. That would be the end result of all of your hard work. What you're going to end up doing, you have to recognize, is that at each one of these spots, you're going to have genomic clones that do this and all kinds of different things. And you got to figure out which one is the best clone that is extending your walk in the correct direction. And it's another PhD project back in the 80s. But as you're doing this, you're taking the genomic DNA and you use that to look at RNA. You use it as a probe for RNA blots. So if you're working with a gene that's expressed in the fly brain, you'd be extracting RNA from the fly brain or from the head, and you'd look to see, is there a gene here that is actually going to hybridize to messenger RNA from the brain? And if it does, is it something that's specific to the brain? If it's, does it make sense in terms of what the mutation is doing? And you can identify candidate genes and then begin sequencing them. Another classic analysis tool, once you've got a gene identified, is another kind of in situ hybridization. Rather than doing it with chromosomes, we can do an in situ hybridization, and this is going to give you the tissue localization of your gene. You're asking the question, where is the gene being expressed? Now, I guess that should be a question mark. So you can have a experiment in which you can say, okay, we know it's expressed during early embryogenesis, or we know it's expressed in the adult. But if you really want to get down to the better detail, where in the embryo is it being expressed? Is it being expressed in the developing nervous system? Is it being expressed in the developing muscles? And if it's in the brain, what part of the brain is it being expressed in? So these are some of the overall tools we can begin using to actually understand what the gene may be doing, where it's being expressed, its location, and the ultimate sort of combination is to have a mutation, to have the genomic clones associated with that mutation, with that gene, and cDNAs associated with that gene. And if you got that, there's all kinds of experiments you can carry out. So from this, we can begin to carry out really good genetic analysis about how genes are working and exactly how these different kinds of mutations are affecting the gene product and things like that. So we're going to stop here. I'm going to put together a DNA sequence analysis experiment for you to begin looking at. I'll have that for you for next week. That's all I know. I'm out of here. Bye.